Donc I would like to thank the organizer for giving me the opportunity to present today this result. And I will speak about the involvement of NUP15 in ALS. As you know, there are many different pathways that are altered in ALS, but in recent years, there are many studies that show that nucleocytoplasmic transport alteration are a key event in ALS pathology. So it can affect the importin or transportin that are transport receptor, or it can affect nucleoporin, and its protein can present a reduced expression or mislocalization or cytoplasmic aggregation that lead to nucleocytoplasmic transport alteration that promote protein mislocalization. And in the lab, Salim, by a genetic analysis, identified different NUP50 variants that increase the risk to develop ALS. And NUP50 is a nucleoporin present at the nuclear pore in the basket, and is also it can be also diffused in the nucleoplasm since it's a mobile nucleoporin. And all the variants identified by Salim are in the N terminal, except for the one variant that is present in the FG repeat domains. And most of these variants are predicted as highly pathogenic. So the purpose of this project is to analyze how NUP50 variant increase the risk to develop ALS. And for that, we study the stability of the protein, its localization, and its interactome. And we also tested this variant in zebrafish. And to study the localization of the protein, we generated different plasmid with the wild type of the variant form, tagged with a ASHA tag, and we overexpress them in neuronal cell type. And by Western blot analysis, we did not observe a difference in the stability of the different variants. Then we analyze the localization of this variant at the NPC, and for that we quantify the percentage of colocalization between NPC marker and NUP50. And as you can see, we did not observe a mislocalization of the different variants, and the percentage of colocalization is similar in all the conditions. We also analyze the localization of NUP50 in the nucleoplasm, and for that we quantify the ratio of the fluorescence present at the perinuclear region and in the nucleoplasm. And as you can see here, we did not observe a difference for this different variant of the localization of in the nucleoplasm, and the ratio is similar for all the variants. Then we analyze if this variant can affect the interactome of NUP50, and for that we overexpress different plasmid, and we focus only on 3 variants, one structural variant and 2 missense, and we overexpress in 2 cell types, one neuronal and one non-neuronal. And after immunoprecipitation, we identify the interactant by mass spectral analysis. And first, we analyze the interactome of wild-type NUP50 since it was not described. And we observe that NUP50 interact with 3 different groups of protein. The first is the nucleocytoplasmic protein involved in the nucleocytoplasmic transport. And we also identify protein involved in ribosome biogenesis. And in XX, we also identify a third group that is involved in chromosome organization. And finally, we observe 26 common interactants between X-cells and Neuro2 X-cells. And most of these interactants are protein involved in nucleocytoplasmic transport or in ribosome biogenesis. We also identify 2 proteins involved in simulation and in chromatin compaction, and 1 kinesin. Donc, we focus the effect of the variant on the 26 common interactants, and for that we perform heat map. Here we have the 2 variants, the Y156C and the K275E, and here the variant R45C, and here the wild type. And we can see for the both variants here, we can observe an increase of the interaction, and for this variant, we observe a decrease of this interaction. The most affected protein are the protein involved in the nucleocytoplasmic transport, and we did not observe a difference for the protein involved in ribosome biogenesis. And we also see that the variant present different effect on the interactomes, since this variant induce an increase of the interaction, and this variant a decrease of the interaction. So to see if the difference of interaction with the protein involved in the nucleocytoplasmic transport have a functional consequence, we tested the localization of fluorescent reporters that contain nuclear localization signal and nuclear export signal. And this, with the M-Sherry tag, this reporter can shuttle between the nucleoplasm and in the cytoplasm, and we overexpress with this different variant. And we measure the ratio of the fluorescence in the nucleoplasm or in the cytoplasm. And we see if we overexpress this different variant, these three variants induce a decrease of the fluorescence in the nucleus that suggests an alteration of the nuclear import, or a decrease of the nuclear import, or an increase of the nuclear export. And then we tested the overexpression of these different variants in the zebrafish model, and for that we developed collaboration with Kabashi Lab, and Juliana performed this experiment. You can see if we overexpress a 50 wild type, we didn't have a difference in the distance or in the velocity compared to non-injected animals. In contrast, if we overexpress the variant, we observe a decrease of the distance traveled for the three variants, and for the variant Y156C, a decrease of the velocity. And we also observe a decrease of the motor neuronal axonal length for the three variants, as you can see here. And we observe for the Y156C variant a decrease of the cell body that suggests a toxicity for this variant, and we did not observe a toxicity for the two other variants. So to conclude, we observe that NUP50 is involved in three biological processes that are nucleocytoplasmic transport, chromatin organization, and ribosome biogenesis. And we observe that the variant did not affect the stability or the localization of the protein. However, it affects this interaction with its partner. We see that the variant present different effect. For one variant, we have a decrease of the interaction, and for both variants, we have an increase of the interaction, excepted with the kinesin. We also observe that this variant induces an alteration of the nucleocytoplasmic transport, and in the animal model, in zebrafish, we see a motor alteration and reduced axon length. And for the variant Y156C, we observe a toxicity and reduced velocity. So we are doing this experiment in human motor neuron obtained after differentiation of iPS cells, and we focus our studies on the viability and in the nucleocytoplasmic transport. And in the future, we would like also to analyze the effect of this variant on the genomic function of NUP15. So I would like to thank all the persons involved in this project, and more particularly Olga, Isabelle, and Aurélie that performed this experiment, and also Juliana for the experiment in zebrafish. And I would like to thank all the foundations that are funding this project, the ARSLA, IFM Téléthon, and the Packard Center. And thank you for your attention.